Hello. Now in this module, I'll be looking at the second part of the structure of DNA. Okay. Now let us look at the polarity of the strands. Okay. Yes. Oh, fine. Now look at this pentose sugar, right? Now this pentose sugar, if you recollect, has a nitrogen base on the first carbon, and it also has the phosphate group on the fifth carbon, and this is what is called as a nucleotide. Please recollect that bond between the nitrogen base and the sugar is also called as glycosidic bond, and the bond between the sugar and the phosphate group is also called as sugar-phosphate bond. Now this nucleotide is attached to the next nucleotide with the help of phosphodiester bond, right? There you go, phosphodiester bond. Then to the next nucleotide once again by phosphodiester bond. Then to the next nucleotide also by a phosphodiester bond. So what you see here is one strand of DNA, right? Which is nothing but a long chain of nucleotides attached to each other by phosphodiester bonds. Correct? Yes? And that's a free OH group at the third position. Now the other strand will be facing in the opposite direction. That is what Watson and Crick discovered, which means what you see here is that the sugars are turned upside down, right? As a result of which the OH free group is on the top and the bottom free phosphate group is basically at the bottom, right? Now let's add the H bonds. Now, between A and T you will see two hydrogen bonds. So on the top A and T, and at the bottom T and A, you can see two hydrogen bonds. Since they are weak bonds, I'm going to indicate them with dotted lines. Okay, and between C and G there are three hydrogen bonds each, correct? Right? So this is how a typical structure of double-stranded DNA would look like. Now look at this first nucleotide on the top left-hand corner. I have marked the 5 carbons, 1, 2, 3, 4, 5. Now here the fifth carbon phosphate group is absolutely free, and this end is now called as the 5' end. At the bottom of the same strand, where I'm showing you the 5 carbons again, what you see is that the third OH group is sitting free. Now this end with the third OH free group is also called as the 3' end, right? Now look at the other strand, the complementary strand. Here on the top right hand corner, what you see is that the free OH group on the third carbon basically will be representing the 3' end, while the fifth carbon free phosphate group will be representing the 5' end, correct? So what you see here on the first strand, 3', 5' direction, we'll call as the ascending strand, and on the next strand, what you see is 3', 5' goes down in the descending manner. I'm going to call that as a descending strand. Now since the two strands run in opposite direction, they are also called as anti-parallel strands, right? So the other way of saying anti-parallel strand would be one strand running from 3' to 5' direction, while the other strand runs from 3' to 5' in the opposite direction, hence anti-parallel, which also means that 5' of one strand is close to the 3' or the other strand, and vice versa. Is it okay? And this is meant by the polarity of the strands, correct? Okay, moving on. What you see here now is the point number 6, which is major and minor grooves. Now once again you're seeing at this double helical structure, typically the two strands undergo something called as right-handed coiling. Is it okay? Right? And what you also see, because of the coiling, you see formation of a major groove. What you see here is a major groove. The major groove is also the deep groove. Is it okay? So these arrows basically indicate major grooves. And alternating the major grooves are twisting, which is basically going to form minor grooves. Okay? Right? These are minor grooves. Fine? Minor grooves and major grooves alternate each other. Minor grooves are also called as shallow grooves. Okay, please remember that. Moving on, dimensions of the DNA. Now if you see the DNA typical structure, what you see here once again is the DNA molecule, correct? Now remember, 1 nanometer is equal to 10 Armstrong. So at some places I'll be writing in nanometers, while other places I'll be writing in Armstrongs, because you can get asked in either way. Now diameter of DNA is about 2 nanometers or 20 Armstrongs. Now when I say the word diameter, which means distance between the two strands, it is roughly about 20 Armstrongs or 2 nanometers. Is it okay? Now one turn or one spiral is about 3.4 nanometers or 34 Armstrong. One entire turn right? There will be 34 angstroms, the length. Is it okay? And it's also seen that there are about 10 base pairs per turn or per spiral, and therefore distance between two successive base pair would be close to 0.34 nanometer or 3.4 angstroms. Is that okay? Correct? So once again, diameter is about 20 angstrom, length of one turn is about 34 angstrom, and distance between two successive base pairs is about 3.4 angstroms. Is that okay? Also, there is a deflection of pitch angle of 36 degrees between two successive steps, and that is why DNA takes shape of a spiral helix. Is that okay? And that brings us to the end of the module where I discussed the second part of the structure of DNA. In the next module, I'll be talking about the semi-conservative replication of DNA in several parts, starting with part 1. Thank you.